is back with the CFARs and one of our TB experts to introduce our speakers for the next session. So, welcome. Thank you.
Okay, so it's great to be part of the CFARs after these many years of wandering in the wilderness. So we're back and very happy to be back. And thank you for sharing the anxiety of keeping the speakers on time. So I think that's the real reason that I was asked to moderate the session. So the next speaker is Hulin Wu from Rochester, who's going to talk about rates of TB infection in Cape Town. So,
okay, thank you for inviting me. And I'm the director for the Biomathematics, Biostatistics, and Bioinformatics core in the University of Rochester CFAR. And so I'm a statistician, so do not ask me the hard question on immunology or whatever. And actually, I worked as a statistician for ACTG network for 7 years before I moved to Rochester 10 years ago. So today, actually, I'm trying to share some experience, how statisticians need to be involved at the beginning of the study. As you know that, statistician or computational scientists start to, contribute a lot to HIV research. I think when we try to start to work on TB, HIV co-infection, probably it's a good idea to get more statisticians or quantitative scientists to be involved. And so today I will share this study with. So this study actually is a collaboration with one of our international collaborators. As several speakers mentioned, actually is the Hulin Wu's group, in South Africa. And so as you know that in South Africa, TB prevalence rate is quite high, and there are 70. I'm not sure this is true number. 73% co-infection with HIV. In particular, it's very high. It's in the the high prevalence in TB HIV co-infection in those crowded African townships, in South Africa. And actually, the goal of this study is try to determine the age-specific prevalence rates of latent TB among HIV-negative individuals. Why it's try to figure out this for HIV-negative individuals? Because we are trying to figure out why there's a high co-infection of HIV and TB. Without HIV, do they still have a high TB rate, prevalence rate? And also try to figure out the annual risk and also force of infection during the childhood. The design of this study, actually, this is the setting of crowded townships of Cape Town, South Africa. And this is from a cross-sectional survey using standard skin test. And actually, for the subjects between 5 years old to 40 years old, and we define the TST diameter is greater than 10. Is defined as the latent TB. I think this is a standard criterion to do this. And basically, this Cape Town picture looks very beautiful. If you visit there, I visited there for 2 weeks. I started collaborating with the people there. Actually, some area of the Cape Town, there's very poor townships. This is one of them. These very poor townships. They're very crowded. A lot of people over there. And in these townships, actually, the HIV TB co-infection rate is so high. So that's why these studies, they do this survey actually using the mobile lab. Actually, they drive around the township and randomly sample the subjects. And so in this study, actually, in total, is sampled 1,061 healthy individuals and 832 children, from 5 to 17 years old, and 229 HIV-negative adults from 18 years old to 40. And we use actually statistical methods, which is called generalized non-parametric logistic regression, to model this the pattern of the epidemics. And here is the results. When we look at the result, this skin test results, that we can see here. This the first one is that there's a 0, means it's negative. Then we can see the distribution for those positive of the the skin test diameter. if we define the 10 or more is TB, latent TB, that's, this is the distribution, and the peak is actually 16 to 20 millimeters. This is the basically distribution. Actually, there are 45% of the subject actually, if based on this threshold, 45% actually is latent TB positive. And when we look at the diameters versus the age, there's no trend. Basically it's a flat line. If you see, there's a kind of random. When we look at the TB prevalence trend along the age of the subjects, and we can see the curve of this, and actually this this prevalence rate increased from 28% at 5 years old up to the peak at 31 to 35 years age. Actually, here that the peak is almost 88%. And when we look at the age, gender difference, the dashed line is male and the solid line is female, and you can see the difference is not significant. But when the people are older, that looks like some kind of difference. And the male actually start to plateau, but female start to going down. After this 31 years old. So that's the kind of trend. Here is the prevalence rate change per year. Prevalence rate change per year. Here we can see that actually the prevalence rate change actually is peaked at about 13 years old. Here is that means. So for those kids, probably they have more close contact at this age. That's why I probably have a peak over there. When we look at annual risk, basically the annual risk is between 3.9 to 4.8%, and the force of infection actually peaked at 15 years old. Is 7.9%. This is the table, and this is show the force of infection at different ages. And this is the annual risk. It's basically the 3.9 up to 4.8. So that's the quite high risk. In summary, actually, before the school started, before the kids go to school, actually 20% of them are already latent TB. And by the age of 15 years old, this 50% of them are TB, in these townships. And by the age of 25 years, this is the high, HIV prevalence rate peak. Is 75% of them are latent TB, and also the rate of increase in latent TB prevalence peaked at age 13, as I mentioned earlier. Maybe this age is easier to get TB, and annual risk for 5 to 15 years old people is from 3.9 to 4.8%, and the force of infection is 7.9%. So actually, when we look at this data, compared to the survey data 50 years ago, that's 50 years ago, the medical conditions even was very poor, and actually this S-shaped curve is quite similar. I show you the trend. It's very similar, and also the extremely high rate of latent TB in children actually is as high as 50 years ago. 50 years ago, as I just talked about it, and also from this data we can see that the high prevalence and also force of infection in children before they get HIV probably is a key factor underlying the explosive HIV associated TB in epidemics. So that's the thing. And finally, I just want to mention that our the biomathematics, biostatistics, and bioinformatics core at the University of Rochester, we have the expertise to do kind of competent modeling at epidemiological level, even clinical level, and animal level, and also bench side. In terms of design a study and do competent modeling. So we actually share these resources with other CFARs. This is like 2 studies we collaborated with actually one of the investigators over there. It is from Brown University, and these 2 studies. One study is we work together on the viral dynamics, HIV viral dynamics for HIV-TB co-infection. This paper is published, and another ongoing study. Our statistician helped to design the study to look at antiviral drugs and also anti-TB drugs, PK/PD and also efficacy for this clinical study. So this is finally like the knowledge. Our collaborators, Robin Wu's group, there's several people work with us on this study. Actually, they collect all this data, and also my colleague Hua Liang helped me to do the analysis, and also our DC farm. I think that's what I have.
I was very curious to see the peak incidence of TB at around age 13, and I'm wondering if you have any idea why that would be.
We think probably those kids are at the years of 13, right? Mm-hmm. And they play with it at a very, very close contact. That's the potential reason. But
Can I offer an alternative to think about? This is cross-sectional data.
Yeah.
So it's a one-time survey done recently.
Yeah.
And so 13, what happened 13 years ago? HIV entered Cape Town and rates of TB went through the roof. I mean, in a cross-sectional study like this, you can't really look at the prevalence of TB in the community, over time. But clearly those younger kids have been exposed to a lot more TB in the last decade than older people were when they were kids. So,
Yeah.
but that could have multiple effects on the shape of that curve. But that is, it's curious that 13 years ago the HIV epidemic hit Masipumalela and rates went through the roof.
Right.
Fred?
I think on one of your slides you pointed out that the adults were known to be HIV negative. Did you do HIV testing on the children and adolescents?
I think so. They did all. Yeah, I think all the people are negative, right?
Okay,
Yeah.
'cause it wasn't stated.
Yeah.
I was wondering, do you have any IGRA data, particularly in the young children? Because I'm curious to know if some of the TST positives that you're seeing could really be BCG.
You mean what data? You mean the?
Either at least spot or interferon gamma.
No.
TB specific.
No, I don't think they have those data. They just randomly collect sample. I think they use the candidates, whatever, track those kids, go there to get blood draw and do test random.
No? Okay. Thank you very much. All right, so next we have Nicola Zetola from the University of Pennsylvania. We're going to talk about HIV and social networks and TB in Botswana.
Well, thank you very much. More than a presentation, this is an invitation. You will see the data that we're trying to generate here, and the system that we're trying to generate. Eventually, it will produce a lot of data that can be used for multiple purposes. About 2 or 3 years ago, when we got a developmental grant, actually with Ron being my mentor, so everything is thanks to Ron, and all the faults, everything's also to Ron. It funded two main projects. One I'm not going to be talking about, but I just want to make you aware. We have a prospective cohort of about, so far, 550 drug-resistant TB, MDR TB and XDR TB that are followed on a monthly basis, and it includes every single patient in the nation. Every single patient enrolled in treatment in the nation. So we have it's a very well characterized cohort for in clinical, microbiological. All the samples are stored, sociological, and so on and so forth. But so that is a resource that is available. But I'm not going to be talking about that. So. What the other part that this developmental grant supported was this study. Just to give you a very brief background, Botswana, it is in sub-Saharan Africa. I got there, and I'm living there for 4 years now. Before I went there, actually, I didn't even know where was it, so I'm putting this picture. But it's a great place for many things. One of those is research, because it's been said that it's the size of Texas. I don't know. People has a different accent, but otherwise, it has a very relatively manageable population. 2 million, the entire area. TB rates are overall relatively high, and TB HIV co-infection is extremely high. HIV in the population is about 18%, and TB HIV overall is about 60 to 70%, depending on the numbers that you're looking. So when I got there, I came with a little bit of a public health background, trying to just implement interventions, and infection control interventions you need to know what's driving the epidemic. We realized that since the prevalence of HIV was so high, the paradigms that were available and that are available and are standard up until now may not be applicable. Those paradigms say that transmission depends on, for transmission to occur, in this case needs to be in close and prolonged contact with the exposed patient, and that most of the new cases are actually secondary to reactivation. This is not anymore. These have been just proven to be wrong and turned to be wrong in that particular setting. But this is still unknown, and is still the paradigm that is in place, or that is available right now. And most studies in terms of the TB transmission were from before the HIV era and before the MDR era. So we decided that if we want to implement any infection control interventions that are going to be efficacious, we need to understand what are the factors driving the epidemics in areas with a high HIV prevalence. So looking at this is Botswana again, looking at all the Botswana, we also identified two different patterns in terms of the epidemic. In Gaborone, which is the capital, is primarily urban, and the population tends to be aggregated and highly mobile within the area. HIV rates are extremely high. In the core of the city, there are around 40%. Outside the city, in extended areas, are about 20%. And the HIV-TB co-infection rates are extremely high as well, up to 80%, depending on the numbers that you look at. On the other hand, Hansi is a primarily rural area, and this is where it is not the politically correct word, but the Bushmen, the San, were relocated. These hunter-gatherers that were in the Kalahari were relocated in this area, so they are relatively new to TB. They have not been exposed in the past to TB. They are actually one of the two groups, together with the Pygmies, that are genetically more homogeneous in terms of their human genetic background, and they are in this area. And because of their social behaviors, they have HIV rates that are extremely low, or extremely in a sense, because it's 10 to 20%, but most of these 10 to 20% is people that have migrated from here to here. They tend to be located there, not mixing with other populations, and the TB rates that are not associated with HIV are extremely high. So it's over 1,500 per 100,000 people. So these are clearly, or seem to be, very different epidemics, at least patterns of epidemics, and allow us to study what is the effect of HIV and drug resistance in these settings. So our hypothesis was basically that there are two different epidemics and that they are fueled by different mechanisms or driven by different mechanisms particularly HIV and drug resistance. And what we actually believe, and for people that works in the in areas with high HIV prevalence, most of the patients with TB cannot tell you who was the contact. So we believe that a casual contact, either in a combi or waiting for a few hours in a clinic or something like that, is enough for an HIV positive patient to develop or to acquire TB, which is completely different than in immunocompetent patients that require prolonged, sustained exposure. So the main aim of the study is basically is a very simple study, very complicated logistically. You will see. It is to determine what is the effect of HIV infection and what is the effect of TB drug resistance over an epidemic, TB epidemic. In addition, because we're going to be collecting all the samples, we want to see whether there is a differential distribution of the strains, whether drug-resistant strains, less fit strains, are more prone to be in certain populations, like in HIV positive or nosocomial populations, or so on and so forth. To identify the hotspots for transmission and to identify these spots to interrupt the chain of transmission. These are kind of mini outbreaks. And to identify previously unrecognized areas of or social networks where TB is taking place. So 4 things are going to be key for this study to be useful and successful. The first one is we're going to compare whether one of the main things is in an area with HIV positive, high HIV prevalence versus an area with low HIV prevalence, if patients in one area are clustered with the same bacteria but do not have any links epidemiologically, we can assume that those were casual. As opposed to on the other hand, if you have clear epidemiological link amongst people infected with the same bacteria, you can say that the transmission occurred through close contact. So this is one of the things that we're going to be looking, and that is key. But to make sure that you know whether the link occurs, you actually need to identify every single case in the population. So identifying 100% of the cases in the population, it is a must or a requirement for the success or to draw conclusions in this type of study. The other thing, so we need to collect all the cases in the area. All the cases need to be, because the only way to gather the cultures or the clusters is by culturing and genotyping. So we need really good samples. And we need to be able to, everybody needs to be tested for HIV, and we need to collect good epidemiological links. So temporal spatial links are kind of key in that regard. And for that, GPS mapping is going to be key. So the 2 main areas that we're now focusing are these. And right now, we're covering, as you will see, all of these and all of these. And in a couple of months, we'll cover all of this. So we will have a very good population, a very well-characterized population, hopefully with every single case identified through active and passive case finding, in an area that is half of the size of Texas. So everybody will be enrolled. We are collecting all the sputums through induced sputums, and HIV testing is performed in every single case, and all the cultures all the samples are cultured, and all the cultures are genotyped. Data is collected, important data is collected on the first encounter, and then the patients are followed up to see whether they move and how they interact with other social networks. The analysis, well, I'm not going to talk about that, but we have. This is Javarone. In blue are all the clinics, and believe me, before this, there was absolutely no TB research infrastructure. So it is something that, Ron built. All the blue clinics are clinics that provide TB care. The green ones are the clinics that in which we are identifying, collecting, and following up patients. These are our two, and they're the only ones performing AFBs, and this is the only one performing cultures. So we have kind of, in a sense, the monopoly of all the TB that is identified through passive referral in these two areas, and all the everybody with HIV and all the cultures that are collected by our staff. So this is how this is one of our porta cabins. The space was an issue, so we had actually to put porta cabins. Now we're expanding to other areas. They are all our staff has the expertise for HIV testing and TB case management. So they also perform. So in addition to collecting the cases, we're collecting the contacts and their linkages. And something that it's sputum and DOT. Our rates of collection are pretty good. I am out of time, so I am not going to talk about this. But this is so far we have collected 84 or 85% of the entire cases identified in the last 2 months in these two areas, and over 90, close to 95%, had a really good quality sputum, mucopurulent, that is now most of them is growing. All of them have HIV testing. All of them have a good. This is the last one, because GPS is key to establish temporal-spatial linkages is key. So we have actually mapped with good coordinates almost entire nation, at least the entire half of the nation. Each has been coded, and it has coordinates. And categorized in 1 out of 20 different categories. That's where we are. That's somewhere there is me. And